Welcome, good evening, good afternoon, good morning, whichever time that this video has found you in. I want to welcome you. These are Vukanzi tuitions or this is Vukanzi teachings. So Vukanzi is an academy, an educational academy that offers good and quality teachings from grade 1 all the way to grade 12. It has both the tuitions program and the school programs. It's when it was founded, or when it was established, it was known for tuitions. It started by offering tuitions first, then it's moved into now the school programs. So today we have biology. On the program we have Teacher Muvanga. We have Teacher Muvanga or Professor Muvanga. That's my goal, is to become a professor. So I'm practicing one. Yeah. So that's a man who will lead you in biology session today on a very straightforward topic called enzymes. Okay? So wherever you are, we are doing this topic called enzymes together, and then I will show you how exam questions come during the past papers and then see how we can answer and attempt them. Okay, so this is how, let me just give a quick studying technique to the grade 12s out there, to the grade 10s, to the GCs. For instance, when you're studying this topic called enzymes, how can you study the topic enzymes? Trust me, even me, for me to teach this topic, I use some materials to study. So know which material you're using. Is it a past book that somebody that was using for biology notes, school book notes, or a textbook? So you need to have something like the notes either from a normal book, either from a class book. You can have a textbook. You can have a textbook, or you can have a pamphlet, or you can use internet. You can use internet. That's how we study. You need materials. Okay? You need materials. So for me to teach this, I had used a material called Boniface. It's even the one that I will be able to. There is also another material called, not Boniface, but it's just called Simplified Biology. I know as others it's written Simplified Biology Boniface, but there are two. There's Boniface, and the one which is just written Simplified Biology, different from Boniface, but yeah, things are similar. I also use Progress to do this. I had also used Longman, and I had also used internet, which is just Google. So all these have helped me to understand what and my past biology book yakudala yam grade 10 from Nadi, it also helped me to study this. That's the best way you can study and understand. Okay, So I will be able to take you through each and each. So when you're studying such a topic, let's try by all means to start with a definition. Anything you're studying, start with a definition. And definitions differ according to the books that you are using, but they all have similar things. Okay? So the definition for enzymes is very easy. An enzyme. Well, there are some. So now I'll give you practically, like Boniface, it will just say an enzyme is a biological catalyst. An enzyme is a biological catalyst. By the way, be writing what I am saying because I will not be managing to write everything on this screen that you are seeing here. Enzymes are biological catalysts. That's all. It ends there. Okay. So if you read in progress or Longman, it will go a bit further. If you check on internet, it will go a bit further. So materials differ. Others are shallow, others are deep, just like that. Other books will say an enzyme is a biological catalyst that speed up a chemical reaction and remain unchanged. Others will say an enzyme is a biological catalyst that speed up a chemical reaction without itself being changed. I'm saying the same thing, but just using different change on text. So that's how it is. Whichever you pick, I don't mind. I don't mind whichever you pick. They are all materials. But the one I like using it is an enzyme is a biological catalyst that speed up a chemical reaction without itself being changed. Yeah. But either way, yeah, everything is fine here. So when you know what an enzyme is, the definition, because it will come. By the way, I will use Zambian syllabus past paper exams. So if you are non-Zambian and you're watching this, you are welcome, because biology is the same throughout. But when it comes to past papers, we are going to use Zambian past papers. So you pardon me for that, because I want to help those who are sitting for the exams soon. Yeah, but you are free to use your past paper system in your country. Try to know examples, whatever you are studying. So I'm trying to show you studying techniques as well as teach this topic. Know some examples. What are the examples of what you're talking about? We've said an enzyme is a biological catalyst that speed up a chemical reaction without itself being changed. Okay? So now, what are the examples of these enzymes? There are many. There are millions and trillions of enzymes. Some I don't even know. But I'll try by all means to stick to the digestive enzymes, because I feel they are the ones that are cardinal to the Zambian biology syllabus. So we've got enzymes like salivary amylase. If I don't say exactly the term the way it's supposed to be, pardon me. I'm also an enzyme teacher. I'm not a biology teacher by profession. I am a mathematics and chemistry teacher by profession. So I'm just helping out those who need help in biology. Yeah, that's the beauty of being a teacher. We are trained, our mind is trained to teach anything which has context. We can go through, we can read, we can make a lesson plan and teach you. And I'll sound like a biology teacher, because that's what I was trained for, to be open-minded. It's like a soldier. Soldiers are trained to be open-minded. They can do anything. They can mold their body into anything. So am I. So don't be surprised next time if you see me teaching physics. Don't be surprised if you see me teaching chemistry. Don't be surprised if you see me teaching ADMA or hearing me teaching food and nutrition. Yeah, that's Professor Mbanga for you. So the second example of an enzyme, we can give it lipase. We can go on. We've got lactase. Most of them, pepsin. Those are examples of enzymes. Salivary amylase, lipase, lactase, pepsin. We've got peptidase. We've got renin. Pardon my pronunciations when I miss the R and the L. Remember, salivary amylase, lipase, lactase, there's maltase, there's peptidase, there's pepsin, there's trypsin, there's yeah. So all those I wanted to say insulin. So there's what do you call this? There's one I want to say. Now the pronunciation, but all those are examples of enzymes. Remember, I told you that there are many. There are a lot of them. So now, after knowing examples of enzymes, we now need to know what is called characteristics of enzymes. How can we know that we are dealing with an enzyme? Those are characteristics, features, that's characteristics of enzymes, okay? Not factors. Factors that affect enzymes, activities, and characteristics. They are not the same thing. Characteristics, it's feature. Huh? Not they are saying write one factor that affects the rate of enzymes. You are mentioning the protein in nature. How is that a factor? Huh? That is a characteristic. So when you hear characteristics, it's far different from the factor. Number one, all enzymes are protein in nature. All enzymes are protein in nature. I think I have this in Boniface. Let me just go to Boniface so that I show you quickly. Boniface is here. Then I'll just be explaining them. Yeah. By the way, I forgot to mention that enzymes, there are enzymes that can work inside an organism and there are enzymes that can work outside an organism. Those that can work inside an organism are called intracellular enzymes here. The ones that is listed in blue. But you can learn some as well about enzymes. Yeah, I was telling my wife if she can join the meeting. Anyway, she's a clinical officer. She knows this, so it's not a big deal for her. So, ladies and gentlemen, they are called intracellular enzymes. Intra, that is in. Okay, cellular now, those are like cells, okay? Within, inside. Yeah, then enzyme, just a biological catalyst. Those that works outside, they are called extra, meaning outside cellular enzymes. They are working outside an organism. So I forgot to mention that. In case they ask you, please be able to know those things. Then I mentioned about examples of enzymes. This is what I'm just flipping down here so that it can help you. Yes, I mentioned about examples of enzymes. These are the ones that some of them, they are not all of them, but just those that are helpful in the syllabus. There's amylase, there's maltase, there's sucrase, there's lactase. When you hear these, there's a protease, there's peptides, there's lipase. All these are examples of enzymes. In case I ask you, name one enzyme that you know, these are enzymes that are workable. The one that I had forgotten the name, there is Zimes, Isangomfiama baking, Isangomodi yeast. This is an enzyme also. Most of them, they end with the ASE. Okay. There's maltase, which you already talked about. All these were under examples of enzymes. Don't get lost. This is the one that I had forgotten. It's called isomerase. Isomerase. It's an enzyme also, which is responsible for sweetening things. Okay, it changes glucose to fructose. If you want to make sweeteners, you use this enzyme called isomerase. So all these are just examples of enzymes that I am showing you in this good material that I'm using here. Yeah, there is also this one called cellulases and pectinases. All these are enzymes. All these are just names of enzymes. Okay, let's get back to the board. Now that we've known the names of enzymes, remember we started an enzyme is a biological catalyst that speed up a chemical reaction without itself being changed. Are there examples of enzymes? Yes, they are there. There's lactase, there's peptidase, there's pepsin, there's salivary amylase. Now, are there characteristics of enzymes? What does the word characteristic mean? It means a feature. Something that you can use to know that you are dealing with an enzyme. Something that you can use to know that, okay, this is Mr. Mvanga, this is Professor Mvanga. One of my characteristics is that I have a slow speech. I speak slow. And I take my time when I'm teaching. I don't rush. The first one is a protein in nature. So let me just scroll through this so that you now see all the characteristics that we have. They are here. So if this material that I'm using, it has got both characteristics and factors. So now, one of the characteristics is that so here we can simplify. If you are writing out there, you can write what I am saying and not what you are saying. You just say, enzymes are protein in nature. What does that mean? It means that they contain proteins. They are made out of proteins. Be very careful. Proteins are not enzymes in nature. No. But enzymes are protein in nature, meaning that enzymes, they contain proteins in them. And proteins don't contain enzymes. That's one thing you should do. No. Okay? Yeah. So that's what the first statement means. Most of them are protein in nature. So that statement shows that there are some which are not protein in nature. If we to use other materials. Yeah. Then the number two, another characteristics, another feature that you can tell to say this is an enzyme is that they are catalyst. What does that mean? They speed up a chemical reaction without themselves being changed. Okay? An example is myself. I am speeding up a reaction of you understanding biology in a simple way and pass. Am I going to remain changed? No. My results, I got a distinction in biology. So whether you get a distinction in biology, whether you get a credit, a merit, or you get a fail, my results will remain unchanged. So I'm a teacher who is acting as a catalyst. So that's what it means. They just speed up a reaction and without themselves being affected. Number 3, they catalyze both forward and reverse reactions. So for the forward reaction, these are the arrows here that I'm viewing here. They are the ones that we used to do that. And then this one, take note of this one. They are specific. Do you know what that means? This enzymes are specific. The enzymes that can work on proteins, they can't work on starch. The enzymes that can work on starch, they can't work on fats or lipids. That's what it means. Just like a lock and a key, they are specific. The key that can open the kitchen door may not be the same key that can open the bedroom door or the pantry door, okay, or the storeroom door. So they are specific. They have what they work on. They are affected by temperature and pH. So take note of those things that we've mentioned, ladies and gentlemen. And then we are now going to the properties. The factors. Factors now. Are there, so don't, the characteristics, it's a feature. How can you know that you're dealing with an enzyme? They are protein in nature. They are affected by high temperature. They are affected by high pH or low pH. And they are specific. Don't forget that one. They are specific, meaning they work on certain things only. Now, factors is what can make them to be slow, what can make them to be fast, or what can make them not to work. That's factors. Something that causes you to do something. Not that identifies you. That's a characteristic. So number 1 is temperature. Do people do physics? Don't think wherever there is temperature, you have to say pressure. Under enzymes, there is no pressure. So enzymes are affected by temperature. Whether low or high, they are affected in one way or the other, in a good way, in a bad way. They're affected by pH. pH is potential hydrogen. It is how acidic a solution is. That's pH. So if they say write down 2 other factors that affect the rate of an enzyme activity, you can pick from temperature, you can pick from pH, you can pick from substrate concentration, you can pick from inhibitors, cofactors. All those are factors that affect the enzymes. Remember, an enzyme is a biological catalyst, something that speeds up a chemical reaction. So what can make it fast or slow? It's temperature, pH, substrate concentration, cofactors, inhibitors. All those things can affect. Let me get back to my study material and show you what I am talking about so that you do understand. Temperature is here. How does it affect an enzyme? Does it lower the process or fasten the process? Well, when the temperature is low, the process will be low because an enzyme will not be active. It will be at a low level. When the temperature is getting high, the enzyme starts to become active, and the reaction or the process will be fast. But when it goes beyond, let me do this in a graphical representation so that you do understand what it means. We're talking about temperature. If this is a graph for temperature here, degrees Celsius, enzyme. If here we've got enzyme activity, if here we've got temperature. So when it is low, the enzyme is low. When it is getting better, high, the enzyme is fast. High. The enzyme starts to have high temperature and it will be destroyed. We call that denatures. So there is this point where the enzymes works best here. This point, it's called optimum temperature. Master that word. Optimum temperature is a temperature at which an enzyme works best, okay, of its ability. Once it crosses here, it starts to become inactive. The shape will be destroyed. The shape of the enzymes will not be the same. And once it reaches somewhere here, it will be denatured. Actually, but once it exceeds, it is denatures. This is what it means when you hear that the enzymes denatures. That enzyme activity increases with increase in temperature up to optimum temperature. Take note of this in blue. Optimum temperature. It is a temperature at which an enzyme works best. You look at the number. So whatever the number that is here is the answer. If it is 28, then the answer, if they ask you about optimum, you say 28 degrees Celsius. Okay, whatever number it is here, that's the number that you pick. Optimum temperature. Yeah. So that's what it means. Take note of this word, denatures. Now move up. At optimum temperature is a temperature at which an enzyme works best. Denatures here, it means the enzyme has passed the optimum temperature. The temperature of an enzyme has gone beyond optimum and it is destroyed. Denatures. The other one, ladies, so this is the graph of optimum temperature where they are dotted lines. That's the optimum temperature. Sorry for that. There's pH. What does pH mean? Potential hydrogen. It is how the amount of acid in a solution. So when the acid has too much solution, it will affect an enzyme. Too low acid solution, it will also affect an enzyme in one or the other. So here on pH, we'll say there are enzymes that works well in mediums that have no acid, and there are enzymes that works well in mediums that have acid. Okay, what are we saying? What are we talking about? When you look at, there are regions in the body mostly. So the first region is the mouth. The mouth doesn't contain acid, it contains what is called alkaline. Number 2, there's a region called stomach. There is duodenum, there is jejunum. So the stomach is acidic. Duodenum is alkaline. Okay, duodenum is a bit mixture. Jejunum is alkaline. Iri mixture plus muri acidic. So there are enzymes that can work well in the mouth, others they can't work well in the mouth. There are enzymes that can work well in the stomach, others can't work well in the stomach. There are enzymes that can work well in the duodenum, others can't work well in the duodenum. There are enzymes that can work well in the jejunum, others can't work well in the jejunum. What are we talking about? You've heard of salivary amylase. It works well in the mouth. Why? Because there is alkaline. But it can't work in the stomach because there is no alkaline, there is acid. What will work here is pepsin. Duodenum, both. Jejunum, mostly alkaline. Okay. Also both. So that's what it is meant by pH. Graph. So you have to master the graphs. Okay? It has a graph as well. It goes like this. Here it is called, at this point, it is called optimum pH. Optimum potential hydrogen. That's what it's called. If they ask you about this number, you read it and see what happens. Well, I haven't researched on what happens if it goes beyond the pH. Okay, so that's your homework. I'm also giving you. All researchers find out for temperature. If it goes beyond the optimum temperature, it denatures. So find out on Google, find out in other books what happens if it goes beyond the optimum pH. Okay, so now we are almost done. Before I talk about, let me talk about uses of enzymes. Enzymes has got uses. We are clocking uses of enzymes. Enzymes can be used in baking. So you need to explain how in baking. Enzymes can be used in brewing. How in brewing. Enzymes can be used in sweetening, making sweeteners. Philadelphia lower and if my juice, if my shine, not even if my fever pangira kwa abana if my sweet shine. Enzymes can be used in juice making. Juice making, washing paste or powder, soap. All those are uses of enzymes. So the homework that I am giving you is to know how to explain how is an enzyme used in baking, which enzymes is used in baking. So take note, which enzyme is used in baking, how is it helpful in baking, which enzyme is used in brewing, how is it used in brewing, sweetener, how is it used in sweetener, juice making, how is it used in juice making, washing powder, how washing powder, and there's what is called tanning leather. How is it? I'll beam so that you read on your own. I'll beam for you the notes. I'll provide the notes. And then it's just you to read and master how it is used in all these. Okay? I'll do that shortly. Let me talk about now some terms to know before we wrap it up. Some terms to know. They are very important terms to know. These are very important. Number one, you should know a term called substrate. Number two, you should know a term called active site. Number three, you should know what is called, let me do this up if you've taken note. Number three, you should know what is called substrate, enzyme substrate complex. Then number 4, products. Once you know this, it will be very easy for you to understand enzymes. So let's start with number 1, substrate. What is substrate? It is a substance that an enzyme works on. Simple. A substance that an enzyme works on, or a substance that an enzyme speeds up. You can also put it that way. Examples, there are many. Number 1, we've got carbohydrates. Carbohydrates, or we'll just say starch to shorten it. Number 2, we are under substrate. What is a substrate? A substance that an enzyme works on. That's a substrate. Examples, carbohydrate. If you have the food that we eat, they are substrate. Okay? There's carbohydrate, there's proteins. Those are substrate. Okay? There's fats. Those are substrate, or lipids if you want. You can put it that way. Yeah. Navimbi. So those are the ones. If we break it a little bit, we also have, for instance, we can also have maltose. If we break it a bit, this is a broken down of starch. Okay, it is also a substrate. If we also break it, peptides. This is a broken version of proteins. For glycerol, for fats and shan. So this is another examples or other examples of substrate. Hope you got that. Number 2, what did we say? Number 2, we said we have to talk about active site. Active site. Active site. Well, an active site, it is where a substrate fits on an enzyme. Look, if this is an enzyme, I'm just giving an example. If this is an enzyme and then this is a substrate now, so this substrate will fit in here and it will look something like this. It will look something like this. So we are saying this is an active site, a region where an enzyme, or a region where, not an enzyme, a region where this substrate fits exactly on an enzyme. It is called an active site. The rest of these parts where it doesn't fit here, they've got another term I have forgotten, but I'll show you in the notes what they are called. I'm just interested on this part which is called the active site. So an active site, it is just a place on an enzyme where the substrate fits. Number 3, we said enzyme substrate complex. Well, enzyme substrate complex, it is just when an enzyme and the substrate mix. Yeah, like this. So this is a substrate. This is an enzyme. Yeah, so this is complex, like physical complicated. Complex. The addition of an enzyme and the substrate together is a complex. Okay, that's all. So basically, it is just when an enzyme and a substrate they meet, vyakumana. When a substrate fits into an enzyme, that's this part, this node, this it is substrate, enzyme substrate complex. Number 4, actually there's supposed to be 5. The last part is lock and key theory. Number 5, it is products. Products are just results of the enzymes substrate complex. We are saying if this is an enzyme, we're just giving an example. If this is an enzyme with its substrate, what will be the result? See, we need products. Product is results of what was, being formed. So products, you can have something. We're talking about, has this thing been broken or not? If yes, maybe we see something like this: 3 things, 4 things, plus the enzyme itself. Enzyme doesn't change, it will still remain the way it is. So what you have here, it is the product. Let me give a very practical example. There's there's starch. Okay, so starch is an example of a substrate. And then now we've got an enzyme called amylase, salivary amylase. So if this is salivary amylase, SA, not South Africa, but salivary amylase, and then if this is starch, if starch is something like this, so if starch ingayaingira apa kwa, if starch fits here, we'll call this enzyme substrate complex. Okay, this we need products. Will starch be broken, yes or no? If it's a yes, then it is going to be broken into a product called maltose. Maltose varayitova mpa ndimbe muri fibiri. But salivary amylase will remain the way it is. It won't change. That's what it means. Or we can have this maltose being broken by an enzyme. If this is being eaten, we need a product, maybe to aratovika, so whereas this will remain the way it is. The enzymes always remains. And what will be the product called? Glucose. So that is just a sequence. Another sequence I can give you is proteins. When enzymes works on protein, it is being changed to peptides. When peptides are being worked on an enzyme, when an enzyme works on a peptide, it's being changed to amino acids. Amino acids. So what are we saying? We are saying proteins are broken down to peptides. Peptides are being broken down to amino acids. That's a final, that's a product. So the question is, which enzyme works on proteins? Well, one of them is pepsin. We're not going to go in detail how is pepsin formed now, on this topic. What enzyme works on peptides? It is peptidase. Peptidase. And then these are products. Nothing works on the products because you have what you wanted. So don't get confused. All I'm trying to say is if you've got starch, enzymes, they break. That's one thing you should know in layman's language. Starch, they are broken to maltose. That's a sequence. Maltose is broken to glucose. If you want, you can break glucose to fructose, to something sweeter than honey. So what breaks starch? It's salivary amylase. What breaks down maltose? Which enzyme? It is maltase. And then these are products. You can't break products. But if you really want to break down glucose, you use another enzyme. And that's what we are saying. We can't use salivary amylase to break down maltose. No. Each enzyme has got what it works on. That's what it means by specific lock and key theory. Maltose can't work on starch. It has to work on maltose. Just like that. So maltase can't work on glucose. Simple things. The last one, it is lipids, ladies and gentlemen. Lipids are broken down to fats, fatty acids and glycerol. They are broken to two things. So what breaks this? It's lipase. Unfortunately, there's only one enzyme here. It is just lipase that breaks this into two things. That's what it works out to be. Okay, ladies and gentlemen, please don't disappoint me if they ask about the lock and key theory. If they say, can you explain the lock and key theory of enzymes? Don't start explaining about, no, the key that opens to the bedrooms can open the kitchen, pantry. Wow. The lock and key theory, it is of enzymes. It is just a theory that states that each enzyme is specific in nature, meaning each enzyme can only work on each substrate. An enzyme can't work on another substrate which it does not work on. Lock and key theory of enzymes states that an enzyme can only work on one substrate that it works on. Or maybe, yes, no, not even maybe. That's what the theory states. Each enzyme is specific in nature. Therefore, it can only work on one substrate that it works on. For example, I mentioned of salivary amylase. It can only work on starch. It doesn't matter which starch, whether from nshima, from kandolo, from cassava, whichever starch. Okay, whether from spinach, whatsoever, wherever you can have starch from, it will work on starch. Any starch, that's what we mean there. But it can't work on proteins. salivary amylase can't work on protein. That's a lock and key theory. You state that each enzyme is specific in nature. It can only work on one substrate at a time. A substrate that it relates to. All those things should be mentioned. Lipase can only work on lipids and fats. Lipase cannot work on carbohydrates. Lipase cannot work on proteins. That's what it means. Lock and key theory. We've seen students where they say, explain the lock and key theory of enzymes. They start explaining, no, the key can only open one lock at a time. For instance, the bedroom door can only be opened by one key. It's enzyme. These are just theory. You have to relate to biology. So that's how a lock and key theory works. So if this is salivary amylase and I have something like this, if this is a substrate, it won't fit here. Meaning the lock and key theory can't be applied here. It can't. That's what it means. Guys, naranda sana. So I want you to look at the rest of the parts. Now, those are the notes that I mentioned. So if you need notes, I'm giving you all the notes on enzymes. Let me start from where they started from. Yeah, we started from here. This is a summary. We state an enzyme is a biological catalyst that speed up a chemical reaction without being changed. I came to mention about intracellular enzymes, which works inside a cell or inside a living organism. Extra works outside. And then I mentioned about the substrate in the last part of the video. Substrate, I just said the substance on which an enzymes act to form products are called substrate. I also talked about the active sites there, which is where the part of an enzyme where the substrate fits during an enzyme activity is called active site. This is the one I said I have forgotten. Those other parts that I dotted in red, they are called allosteric sites, meaning the parts where the substrate doesn't fit. Here they're just showing you an equation. When an enzyme acts on a substrate, it is going to give you enzyme substrate complex. Then you're going to have an enzymes and a product. We talked about the characteristics of enzymes. We mentioned everything here. So these are notes in case you don't have the notes on enzymes. Explanation of enzyme specification. This is a lock and key. Listen now. One of the theories used to explain enzyme specificity is called the lock and key mechanism. This theory states that each substrate fits into the active site of a particular enzyme in the same way a key fits into a specific key lock, because the two have complementary shapes. The following diagram illustrates the lock and key theory. So this is what I was just talking about. It's about specification. So this is an enzyme, if you can see it clearly. The next thing here is a substrate. So it has fit, called enzyme substrate complex. Enzyme remains the same, doesn't change. Products have been formed. We talked about factors that affect the enzyme activity. We talked about temperature. I mentioned about optimum temperature. I mentioned about denatured. And this is how the diagram looks like for temperature. On top there, it's called optimum temperature. pH, this is the pH. I said the pH stands for potential hydrogen, the acid in the solution. So this is just a pH scale. They are showing you from 0 to 14. If it is 0 to 6, acidic, 8 to 13 or 14, that is alkaline. The optimum pH, we talked about it. This is a diagram for the optimum pH. The enzymes concentration, I talked about it. Those are the notes. Substrate concentration, I talked about it. Inhibitors. So inhibitors, a good example of an inhibitor, it's poison. So anything that will slow or stops the enzyme from working is called an inhibitor. Example, poison. Poison is an inhibitor. It will slow the reaction of things happening in your body. We talked about cofactors, examples of cofactors. So cofactors are those things that don't contain proteins. Vitamins don't contain proteins, and mineral salts, they don't contain proteins. So those are inhibitors. We talked about the examples of substrates, starch, maltose, sucrose, lactose. We talked about names of enzymes, amylase, maltose, sucrose, all those things. And then now, these are the notes that I talked about. They have explained how enzymes are used in making detergents. Boom, bullet, all those things, all those detergent paste that you know. Okay. So here, these notes will explain nicely. Find time to read, because you have to explain something here, how it is used in making detergents. Brewing, you have to explain how it is used in making beer, how it is used in making baking, making bread. There's an enzyme called zymes in yeast. Here, enzymes are used in making sweeteners for food drinks. So you have to know, here it has explained. In daily industry, yogurt, how are they made? Tanning of leather, leather tufala, jacket, shani, leather. It is because of enzymes. There's extraction, juice processing. I talked about juice. This is how it is made. And then tendering of meat. So tenderizing of meat. So all these for baby food. So we are done, ladies and gentlemen. I'm so appreciating that you joined. If you are in Zambia and you need help, online tuitions, this is my number that I use. Yeah, to teach. You can WhatsApp me, you can call me. Otherwise, it's been Teacher Mubanga or Professor Mubanga. I'm not yet a professor, but I will be. But I'm already a counselor by profession. I'm a counselor, so I'm entitled to counselorship. I'm a counselor Mubanga, Teacher Mubanga. I'm entitled, and Professor Mubanga, definitely one day it will happen. I've already discovered some formulas in mathematics. Yeah, so we're yet to be there. Thank you so much. Have a good night, have a good morning, have a good afternoon till we meet again.